Okay, so I start the seminar now. Today I would like to share some information about goose barnacles and the marine forensic science. I'm Jason Hu from Indigo Waters Institute. First, I would like to introduce some Chinese. Actually, it's a local name. Only in Chinese in Taiwan, we call it catch the monkeys. In Chinese, it's called 抓猴. It means catch someone in act of committing adultery or cheating. So if you visit Taiwan, you can see many this kind of advertisements that is related. Advertisement belong to detective agency because people, men or women, want to collect evidence that he or her wife or husband is cheating. So we kind of have these commercials that hire people to catch the cheating. But compared to marine debris, it's hard to catch or find polluter of marine plastic because there's no footprint or fingerprint or DNA on the marine debris. In other terms, it's kind of non-point source pollution. You cannot find the source or polluters when we saw marine debris on the sea surface or on the beach. So evidence is very important. How to find the source or the polluters is one of the topic in marine debris research. And so it's another field called marine forensic science. It's also collect evidence for some areas to use. So first, I would like to introduce, I think it's 2 or 3 papers related to this topic, marine forensic science, and try to find some evidence related to like source or polluters, and most of these relate to these goose barnacles. So this is one of this very important paper in this area, but it's not published in on Marine Pollution Bulletin. It's Forensic Science International. It's called Evaluation of the Floating Time of Crops found in marine environment using the barnacle, Lepas. So the basic story is one dead body was found in Italy at the beach of the small islands, and nobody can recognize the identity of this person, this man. But there's so many small barnacles on the shoes and pants of these crops. So these scientists try to use the size of these barnacles to judge the time of these bodies floating or sinking or immersed on the waters. So the first author is this Paula A. Magnet, a very interesting person. She has some public speech you can find on internet. So this is another research published by this team, because they think the barnacle is a very important indicate. It's not directly evidence or directly indicate, but it's an alternative indicate to show the PMSI post-mortem submission interval, because it's a very important message to this kind of like policeman or judge or this detective information in this field. So they submerge these different sports shoes and these leather shoes into a harbor and to observe the falling animals like barnacle, algae, and other animals in different period. So they found in different substrate, like the maybe it's fabric or the man-made leather the species or the coverage will be different. But it's not related to marine debris. And other papers, these are the published in Marine Biology in 2010. In laboratory, they form these gooseneck barnacles in the laboratory, and you can see in different water temperature, the capitulum length will be, the growth rate will be different. I think I can explain this species first. This is gooseneck barnacle because there has a stalk shaped like a gooseneck, and it's a seesaw barnacles in floating items on the sea surface. And this genus Lepus can be seen almost everywhere in the oceans in the world. So it's a very common wide distributed genus. And usually the scientists will measure the length, the shell length from the tip to the bottom to represent the size of these barnacles. So, and this is a very fast growth species. So you can see in 13 days, this length can reach probably 6 millimeter or almost near 10 millimeters in 50 days. And it's very common when we do the beach cleanup. You will attach on the floating plastic or driftwood, anything, or glass bottles. So there's 2 and 3 papers related to marine debris and these barnacles. The first one is this published by Martin Stay-Fox. It calls minimum drift times inferred trajectory of ghost net found in Maldives. So there are people from Olive Ridley Foundation project. So they are conservationists to the olive sea turtles, but they found a lot of entanglement on these sea turtles, and most of them are a drift net and gill net, this kind of net. But they don't know the source of this net. So they try to use the biofouling, the animal attached on the net, to estimate or to find the source. So they put they put many nets in the waters and to record the animal on the surface of the net during 100 days. So this figure at the right side, you can see these are the days where they put the net into the waters in Maldives. And the y-axis is the average barnacle size. So there will be, at the beginning, there will be a very small Lepus barnacle on this net. But you can see after some days, the barnacle will be bigger and bigger. So these are their experiment. So they use, because these curves are very conservative, and they think it's a very good index to estimate how the net on the sea turtle has been, how long has the net has been drifting in the waters. So they use the sea surface drifting models to estimate where does this net come from. So it can maybe come from the country like Sri Lanka, or country or area from the India Oceans, maybe come from some international fillet or some local fishery from regional countries, because the Maldives is a marine protect area, so it may come from different regions. So they use this equation to estimate the source. So this is one of the application of the this Limpus barnacle. And another tool research, the first one is in Australia. I think I introduced this in our seminar before. So they collect a lot of pet bottles on the East Australia, and they found only 6% with marine growth. So they think probably the source of this pet bottle is come from machine shipping, because that's why there's less marine growth, because there are a lot of shipping or container ships passing this area. So if they dump at the sea, probably they won't much marine growth, because they only drift for a short time and then straight or beached. And another paper is published by Peter Ryan and it's a research in Kenya. So they also collect PET bottle in Kenya's beach, remote beach, and recreational beach, and also on the street, downtown street, and they compare the different measurement, and they also record the marine growth. That means limpus and the bryozoan and some other animals on the surface of the PET bottle. And they say some foreign PET bottles come from neighboring states, but many are probably dumped illegally by ships operating from Asia, because the Kenya is in the south atmosphere, and if Asian bottle, it's difficult for Asian bottle drifting from north atmosphere to south atmosphere. So they have this discussion about maybe it come from ships. So there are some basic information about the barnacle and the marine debris. Okay, so to our this paper, the title is The Ecology of Lepas-based Biofouling Community on Mode Drifting Object with Application for Marine Forensic Science. It's published in Marine Biology. So this is not a marine debris paper. It's kind of marine biology, marine ecology paper, especially on these fouling animals. So I pick up some sentence in the abstract. So we examine the community's succession, growth rate, and isotopic composition in limpus and their associated biofouling communities in coastal waters. So they use abundance of 2 limpus barnacles and the 2 amphipod species create an equation to estimate the minimum duration at sea. So we report a new maximum growth rate for limpus, this species, for 1.45 millimeter per day. Limpus was larger on moored float than free floating drifters. And this data 18 oxygen content of Lepas shells was a robust predictor of sea surface temperatures. So it's an important applications for estimating drift duration and trajectory of marine debris. And they have this infograph on the paper. I think it's very easy to introduce their findings. So they have mooring float on it. So they have many Lepas barnacles, and these are larger if they're moored on the sea floor compared to the free floating debris. The barnacle may be smaller, and it will be predated by some animals like this polycat, the sea worms. So also the growth rate, the growth rate of mooring are faster than is smaller. The species difference, and they are smaller and grow slower. And these two figures here, you can see this is the concentration of delta 18. It's an isotope element. You can see if the temperature, sea temperature is high, the concentration will be high. The temperature lower, it's also the concentration of this element will be lower. And this figure at the right side, it shows the time at sea and the species diversity. So it's when it's in the water, the duration is longer, there will be more different species that live on the debris or buoys. And here, the shoreline scavenging maybe influence community composition. It means that if the debris is beach on the beach, there'll be animals come to eat these animals. Okay, so because I think this paper is quite long, so I won't go too deep on the methodology. So I will combine the method and the result together to introduce to you. So the first is the Bondi Marine Community Ecology. Bondi is a local beach in Australia. So in this area, in this bay, they use this kind of experimental design to use a big mooring buoy attached on the sea floor and with some small settlement plate. So there are some many PVC plate, plastic plate attached here to let the animals to settle down and to grow on the surface of the plate. So they will keep collecting those plates and to analyze the species and the abundance, the diversity on this plate. So in this figure, you can see there are many animals found in this plate, marine animals. Yeah, and there are 4 species of gooseneck barnacles, and some other barnacles, some polychaete worms, and another barnacles, and the crabs, and amphipod, and also sea slug and sea spiders. There are some common fooding animals in the marine environment. Okay, so in this mooring experiment, they find this 36 taxa. Most of them are different species in these 178 days of the whole experiment. And you can see the number of species are increasing from day 0 to day 178. There are more and more species that live on the plate, plastic plate. And they say that there are 3 patterns, 3 both stages on this community. So they use some statistics to represent these 3 different stages. So they collect the samples during the day 18, 15, 84, and these are so on. So you can see the diversity or the composition of the species are shifted from day 17 to day 178. So the composition of species or the abundance of different species are shifting. And these are some figures in the supplementary informations. They are more detailed to introduce these 3 different stages. So in the figure on the left side, you can see from day 17 to the last day. And these are the total percentage of total species. So you can see at the beginning, they are all barnacles. So it's like, just like stage 1. Actually, it's not goose barnacles. They are related species to goose barnacle. This species has no hard shells. They are all soft bodies. So at the beginning, 100% are these barnacles. And in stage 2, there are other algae and some different barnacles. You can see some Lepas attached and some hard shell barnacles here. And at the stage 3, there will be more and more species. You can see some crabs, sea slugs, or green algae, mollusks, and polychaetes on the surface. And they say the biggest cause of this shift probably influenced by predators. And this predator may be caused by filefish. So it's kind of puffer fish. So they have a very strong teeth to eat these hard shell species. So they observe these 2 filefish in these bondy areas. And they also find many shell damages on this settlement plate. So they say at the beginning, there are soft shells and barnacles, but it's eaten by these fish. So there'll be more space for other hard shell barnacles, and also some habitat for different animals like crabs or algae or some amphipod, very small zooplankton or sea spiders. Okay.
Jason?
So yes. Hi.
Maybe I forgot. So could I ask a question for the figure, formal figure?
Yes.
On the left figure, the abundance of barnacles are decreasing. That means the species, the number of species of barnacles are decreasing, or just the percentage are decreasing. I mean, more predators are on the surface, and that means relative ratio is decreasing, or absolute number of barnacles are decreasing too. Percentage. Just percentage are decreasing. It means, for example, 10 species of barnacles are there, but because of the other animals, they are decreasing to 50 species, for example, or just the 100 barnacles exist on the surface, and more other animals are there, then the ratio is decreasing.
Wow. Yes, thank you. It's a very good question. I think actually I cannot answer this question right now, because I think the shifting between stages it's complicated here. They have raw data in the supplementary, like the number of each species in each sampling days. So in some species, I think the author mentioned it's become less and less. I mean, the abundance in some species, it's decreasing now, but in some other species are increasing. So it's kind of the community is shifting, the composition is shifting, and I think they also mentioned the coverage. I'm not sure. The total abundance are increasing. So yes.
Okay. So please go ahead.
Thank you. Okay, so So here, at the result part, the author showed an equation. It says the minimum days at sea can be estimated by these equations. So they introduced four species. The first two are amphipod. It's a very small animals, small arthropod animals, marine arthropod. You can see here this Jesa and this Capybara. And the other two are Lepas species, this Anserifera and this Lepas enitofera. So they say these four species can create equations, but I think it's not represent directly the numbers or abundance of this animal. So they said it represents the code, the ordinal codes here. It means they calculate the area of the plate. The area is 2.16 square meter. So they calculate how many of these species on this plate, and it will equal to some ordinal code. And use this ordinal code in this equation, it can be calculate the minimum days at sea. Okay, so and another experiment is the short-cast debris community ecology. So it's information or data collecting from the beach debris. So sometimes it's maybe plastic debris or some are woods. I think they collect 50 items of short-cast debris on the beaches. And there are some, in this table 4, there are a list of these animals. So these animals are less, the diversity is less. They only found less than 20 species in these 50 items. And the A shows indicate the taxa also observed in the boundary moorings. So you can see some gooseneck barnacle also not only in moorings, but also in the wild beach debris. And there are two results in this community ecology survey. The first one is that the percentage of the size is different. So in this figure, you can see the x-axis is the size of the barnacle, the Capitulian length. So these are larger barnacles. These are smaller barnacles. So you can see if the sample from the short-cast debris, most of them are smaller barnacles. Compared to the fixed moorings, they are bigger barnacles. And another is the surface area. So they also calculate the surface area of these items, use some image software. So they say if the surface area of the debris is bigger, the size of the barnacle will be bigger. So it shows a trend here. And they also mention about there may be many scavengers will eat these animals once they straight on the shoreline, like these crabs, these sand crabs, these beetles, and also some birds will eat the falling animals on the beach. So it maybe influence the data. Okay, so this is another experience called growth rate of lepas, these animals. So they record average daily growth is about 1 millimeter per day, and the fastest is 1.4 almost 1.5 millimeter per day. So it's a very fast growth animals. And they say it's a 5 float higher than the laboratory raised, and the 11 float in similar temperature condition. So in this figure, you can see it's a growth period in x-axis, and in the y-axis is the size of the barnacle. And in their experiment, they are this round dot here. So you can see in a very short time, they are become bigger and compared to other studies here. So this one is very slow here. After 14 days, it's still very small. So they say they record a very fast growth rate. And then the last experiment is a temperature of calcite formation, because this Lepas has a shell, calcite shells, and the calcite is one of the elements of calcite is oxygen. So they use the oxygen isotype, the delta 18, to see the relationship of delta 18 oxygen and the sea temperature. So they will break the tip of their shells to analyze these oxygen concentrations. So in figure B here, they compare the 3 different species, and so they are different, but in this Lepas and Natifera, there are small constraint data shows here, and so they use these species to see in different water temperature. Actually, it's in different season, to see is there a difference of these delta 18 oxygens in their shells. So you can see if the water temperature is higher, actually the change is it's just slightly change. You can see the difference between these elements, and also a difference in different latitude. So they put in different harbors in different latitude, but in the same period, same season, but the water temperature is different. It also shows the different concentration of this element. So they have this equation here, just to use the concentration of this element. They can estimate the sea temperatures. Okay, well, at the discussion part, here they say using a taxa distribute cross pelagic habitat, because this Lepas almost can find any place in the oceans. It's very common. So it can become an estimate of minimum drift times. And the second is the relationship between this delta 18 oxygen content of these Lepas antiphera shells and the water temperature. But the author said it might can, because this is the first research on this isotope of shells, so maybe it can only use in the regional areas. There are some limitations. So there are some application of marine forensic science, like for corpses or weak ships or airplanes. They also mention about the MH 380 from Malaysian Airlines. Also, people also find some limpets on the wings of this airplane, and also marine debris. But for showcase objects the author say it has been noticed that the hard body scavenger resistant taxa may provide a more accurate estimate because these soft body animals can be eaten by those scavengers in very short times. So if we want to find some indicator for shoreline debris, probably we have to choose some hard bodies, different animals as an indicator. Okay, so that's it, my presentation. And do you have